It's a pleasure now to welcome Eric Lander, who's an associate member of the Koch Institute. And you may or may not know this, but Eric actually started out his academic life not in science, but as a faculty member on the Harvard University Graduate Business School, teaching math and science-related business. But during that time, he became fascinated by genetics and worked in a number of different labs at Harvard and MIT to really understand this topic. And then having learned the error of his ways, he then moved to the Whitehead Institute, first as a fellow, then as a faculty member in the Department of Biology, then head of the Genome Center, and now head of the Broad Institute. So all of you know, Eric's name is basically synonymous with all things genomic. He's been awarded numerous honors. I think perhaps the most well-known now is that he serves on the President's Council of Advisors for Science and Technology. And it's a pleasure to have him here today. And he's going to talk about the human genome and cancer. Eric.
Thank you. Well, thanks very much, Jackie. First, I just want to say how excited, both as an associate member of the Koch Institute and as a member of the Broad Institute, whose window looks out on the building going up, how excited we are to see the progress there. And there are all sorts of plans to put in zip lines between the two buildings to have people go zipping back and forth, just clipping on and things. What I'd like to do is talk about just a couple of mostly unpublished stories that relate the human genome and cancer. As Jackie said, I like all things genomic. Why do I like all things genomic? Well, because when you do genomic things, you can take a global perspective. You can see all sorts of patterns that you had no reason to expect in the first place, and they can emerge in different ways. The first things I got excited about with many others was making maps and sequences of the human genome. But in the years since, it's been possible to build more and more kinds of maps, and all of these maps tell you different things. Maps of all the genes encoded in the genome, of all the evolutionary conservation in the genome, of chromatin states of the genome, of inherited variation, of disease associations, of evolutionary selection, and relevant to this particular meeting, to the distribution of targets of cancer mutation. So I like genomic maps because you can find surprises. Now, I'll note that there is, in the world of cancer and genomics, this controversy or tension, which is represented by, most recently, a pair of editorials describing the tension, and it's an important dynamic tension between hypothesis-driven and non-hypothesis-driven research. And of course, the right answer is we need both. It's an ecosystem where data gives rise to hypotheses, and hypotheses cause you to do experiments that give rise to data. So one can't come down on one side or the other of this tension. But I do want to speak, for the next few minutes about the power of large, unbiased genomic surveys for understanding the mechanisms of cancer. So that's the plan. Now, to do that, I first want to just set a little bit of the context of how rapidly genomics has advanced since the time of the Human Genome Project. What has gone on in the last 7, 10 years, depending on what you take as the end of the Human Genome Project. The end of the Human Genome Project was announced on multiple occasions with appropriate ceremony each time, and so it was 7 to 10 years between when, depending on how you count. So let me talk just a little bit about what's gone on since the time of the Human Genome Project and now. So the Human Genome Project was just one big project. It was get a complete map and sequence of the human genome. It involved about 13 years of work of making a genetic map and a physical map and a sequence map and making all that information available and all that. And it took some significant changes in technology. To get 3 billion bases times about 7 or 8-fold coverage, something like 20 or 30 billion bases of information, it required changing the way we do things. So you could make large automated systems so you didn't have to have thousands and thousands of graduate students doing the work, which the graduate students object to, but you'd have machines doing the work and generating a lot of data. It involved, international collaborations between all sorts of groups. And I remember the day that the whole international consortium celebrated the billion base bash, when across the world we had covered 1 billion bases of the human genome to about 7-fold coverage. Great celebration. And then, about 10 years ago this month, the celebration of a rough draft sequence of the human genome, which was actually published about 9 months later in February, and of a complete sequence of the genome published around April of 2003, where complete doesn't mean complete, it means almost complete. There's still about 300 gaps in the sequence of the human genome, but not a big deal. But I always feel obliged to say that there are some important gaps still to fill there. Since then, the world of DNA sequencing has undergone this amazing acceleration. It's been driven, as so many of these things are, by technology. New types of sequencing machines that let you proceed in a massively parallel fashion. From the old days of capillary sequencers and 96 sequences that you would read, these technologies that spread many DNA molecules on a surface and do serial chemistry, adding typically one base at a time and imaging it, or many of the same base at a time and imaging it, but letting you read in parallel and exploiting, as your chemistry gets better and better and your optics get better and better, the tremendous parallelism that's possible. So I want to give you a feeling for what that increase has felt like. This is a graph we used to be very proud of. Is there a laser weapon up here? So, I don't know. Anyway, well, I'll just gesticulate and you'll imagine. This is sequence data produced here at MIT from 1999, when we were so proud we produced 1 billion bases of raw sequence, going up to almost 20 billion bases a year, going up to almost 70 billion bases a year of raw sequence being produced here at MIT through the year 2006. This is what happened with 2 more years of data added to that. It jumps up from 70 billion bases there, whoops, backward, jumps up from there at 70 billion bases, 2 more years, it looks like that, to about 1,700 billion bases through 2008. And then if I add 1 more year to this graph, this is what it looks like. 2008 is down there, and now it's up to 20,000 billion bases of sequence produced last year. And if I add my projection for Christmas this year, it's 100,000 billion bases of sequence by Christmas. And if you ask me about 2011, I think it's 5-fold above that. That's the rate at which this is going. Costs, of course, are dropping. This isn't due to a 10 to the 5th fold increase in budget. This is due, in fact, to a 10 to the 5th fold decrease in cost. Important to say. And what you see in the black line there is Moore's law that's dropping on a logarithmic scale linearly by a factor of 2 every 18 months. And what you see in the red line is the cost of sequencing, which by the end of 2010, by Christmas or so, will have completed a decrease of about 10 to the 5th fold over the course of about a decade. And nothing I know in history has ever decreased in cost 10 to the 5th fold in so short a time. So, all right, what's it good for? Well, it's good for making more and more kinds of maps. And I want to describe 2 different types of maps, and in particular, cancer maps. And I'm going to describe one particular story. And then I'll also talk about chromatin state maps and things we're learning about them that appear to be relevant to cancer, too. And if there's time, I'll mention one other thing. So mapping the cancer genome has been an effort that a number of people around this community at MIT and Harvard, around the Broad Cancer Program, have gotten together around, particularly Todd Golub, Matt Meyerson, Levi Garraway, and Stacey Gabriel. Now, obviously, the idea that the genome is very important in cancer has been around for quite a while. Since Boveri published in 1914 the idea that chromosomal defects cause cancer, there was the little detour of 1970, the declaration that the viral origin of the majority of malignant tumors was now documented beyond any reasonable doubt. One should always be suspicious of statements like that. But within not so long after that, the demonstration of cellular oncogenes and the demonstration by 1986 of all the major kinds of mutations that you would expect. They all can occur: point mutations and translocations and amplifications and deletions. All those examples were known by the mid-1980s. That led, in fact, Renato Dubecco to argue in an important article in Science that a major reason for sequencing the human genome should be for cancer, so that we could take systematic approaches to cancer. Well, in about 4 or 5 years ago, a group, Lee Hartwell and I, and a group on the National Cancer Institute's advisory board, began talking about the idea of a cancer genome project to begin to document all of the mutations that occur with any significant frequency in cancer. It was still technologically a little ways off, but it seemed like it might become possible. Now, there were concerns raised. One concern, all the cancer genes have already been learned. There's nothing more to discover. The other concern was there are so many cancer genes, we'll never be able to make sense of it. One wanted at times to get those two sides to sort of hash it out with each other. But in any case, I'm a firm believer in the middle that we don't by any means know all of the mechanisms in cancer, and yet it's not so infinitely complicated that we can't work it out. So in support of that, what I'd like to do is describe, I'm going to scroll down to right there, but also describe the kind of approaches that are getting used. They are sequence the entire genome of a tumor and a normal and compare them. When you compare a random person's blood to a random person in the population, you'll find, on average, 1 polymorphism every 1,000 bases. When you compare someone's tumor to their normal, you find, on average, 1 mutation every 1,000,000 bases. And you can do this by sequencing the whole genome or by, for example, sequencing any selected part, like the exome, using a capture technology. So I want to describe, I'm just going to scoot over that there, one project that has just been completed and just been written up and submitted on multiple myeloma as an example of the sorts of things that are still there to learn. So multiple myeloma, about 20,000 new diagnoses per year in the United States. It's a malignancy of plasma cells, and it's characterized by massive secretion of immunoglobulins. And that massive secretion of immunoglobulin proteins is relevant, and we'll come back to that in just a moment. So what we did, and we as Todd Golub and Mike Chapman and Mike Lawrence and others, and Gadi Getz, was sequence the whole genome of 23 tumor normal pairs and the whole exome of 16 tumor normal pairs. So the landscape looks like this. There's about 3 mutations per million bases, a little higher than the average of about 1 that you see in many cancers, but lower than what you see in others like melanoma. There's about 35 non-silent amino acid changing coding mutations. And there's about 21 on average chromosomal rearrangements per sample. And you get some genes that have 1 mutation. In fact, most genes have at least 1 mutation somewhere. About 100 genes have 2, 3 genes have 3 mutations, 3 genes have 4, et cetera. Now, how do you make sense out of any of these? Which of these matter in some fundamental way? Well, to sort through a list of mutations, you can look for a couple of things. Genes that on their own, just as an individual gene, have a statistically significant frequency of mutation. One gene has 8 mutations. You do the arithmetic right, yes, that's highly significant. You could look for genes that don't have so many mutations, but you see an identical mutation across patients. Or you could have genes that have a known role already in cancer, using here a hypothesis-driven approach. And then you can also look at not individual genes, but sets of genes, and ask, are there too many mutations in this set? So those are your tricks. For individual genes with statistically significant frequencies of mutation, you have to correct for the size of the gene, the base composition of the gene, set some false discovery rate. And when you do that for this multiple myeloma data set, this is what you see. You find 10 highly significant genes. 4 are previously known, NRAS, KRAS, P53, CCND1. 6 are new. How do we know that these are real? Well, that they're important. One easy way is you can assign every mutation a functional significance based on how much it changes a conserved amino acid and in what way. And it's clear that those genes that have multiple hits in it show much higher functional significant changes. They affect more conserved amino acids or make more significant changes to those amino acids. So that gives you a good feeling that this probably matters. Let's take 2 genes in particular. FAM46C and DIS3, the first 2 of the unknown genes. What are they and what do they tell us? Well, DIS3, there are 4 separate mutations. DIS3 encodes an endonuclease component to the exosome, and the exosome is highly conserved all the way down to E. coli. The exosome has multiple roles, including degrading mRNAs and tRNAs. The 4 mutations cluster there in the picture around one little pocket of this protein, and every one of those residues is highly conserved down to yeast and to E. coli. In fact, 2 of these 4 mutations have been made in yeast and E. coli, and we know their function. 2 of them, just adventitiously, have been made. One of them leads to the significant accumulation of initiator tRNAs. And it's known from separate experiments that relatively modest increases in initiator tRNAs are capable of transforming fibroblasts. The other is known to completely abolish the exonuclease activity. And so the hypothesis here is that DIS3 increases protein translation through increasing the initiator tRNA and through the loss of the exonuclease activity that degrades RNA. So that's a hint that protein translation may be affected. FAM46C, 5 mutations. What is it? If you type it into PubMed, it comes back 0 instances found in PubMed. But if you just take its gene expression pattern across tissues and run it against all other genes, it's a slam dunk match in its gene expression pattern for all the genes involved in the ribosomal protein module. It is perfectly co-regulated with ribosomal proteins. What it does, I don't know, but I'd bet very good money this is involved in ribosomal proteins. And if we take together DIS3 and FAM46C, we also see single mutations in a ribosomal protein gene, ribosomal protein 10, in the ribosomal protein kinase S6, in initiation factor 3A, in XBP1 that is involved in the unfolded protein response, and in which mouse mutations actually cause multiple myeloma, but mutations have not previously been identified, and in LRRK2, a gene known in Parkinson's disease, and it's involved in phosphorylating initiation factor 4E, a binding protein. So there's a whole story here about protein translation and stability that emerge from those mutations, and now lots of hypotheses and lots of work to do. Now, you could also look for any instances where you see exactly the same mutation occurring twice, and that indeed occurs too. IRF4, interferon regulatory factor 4, has mutations in 2 cases, and they're the identical mutation, a K123R mutation, which is a hallmark of kind of a gain of function in many oncogenes. Never been previously reported in multiple myeloma, but recently it was proposed to be a potential oncogene in multiple myeloma based on shRNA screens that knock out the gene and have selective lethality to myeloma lines, but not to other cancer cell lines. Now, IRF4 regulates PRDM1, which is a transcription factor involved in plasma cell differentiation, and 2 of our patients have mutations, this time different mutations, in PRDM1. So there's another story that emerges in there. Then there's just genes that play known roles in cancer. Single instances, but not statistically significant on their own, but you shouldn't turn your nose up at them. BRAF. There's a BRAF mutation in one of the patients. BRAF is, of course, important in other cancers, and the particular mutation that was seen in this patient is a known activating allele of BRAF, although not the one that the Plexicon drug is made against, but one that could be susceptible to other BRAF inhibitors. And when you just go and look now in a collection of additional multiple myeloma patients, and you just genotype them for 12 known activating BRAF alleles, you find that 7% of multiple myeloma patients have these BRAF mutations. So singles can be good. Then there's gene sets. Not individual genes being significant, but a set of genes being significant. And you get your sets in one of 2 ways. A prior hypothesis about a candidate gene set, your favorite gene set, or an unbiased search against the collection of gene sets. Prior hypothesis. Well, the NF-kappa B pathway is known to be activated in a number of multiple myeloma cell lines, and the basis for that is poorly understood, although there are 1 or 2 cases in which TRAF3 mutation is known to do it. So you make a candidate gene set here of 58 genes previously defined in the NF-kappa B pathway. You look, you get a highly statistically significant result with mutations in 10 genes and translocations in 5 more cases. About 39% of patients now have identifiable mutations in the NF-kappa B pathway, suggesting a broader role for the NF-kappa B pathway. Histone methyltransferase pathways. Oops, that should have gone away at the bottom there. It's known in acute leukemias that HOXA9 is activated by demethylation, for example, through MLL mutations. Well, we went and looked, and HOXA9 also shows high expression in multiple myeloma. That is, this is the chromatin silencing of HOXA9, and the chromatin silencing goes away in many multiple myeloma cell lines there, and this is especially true in the cases that don't have the common translocations. Now, the first case we looked at had a mutation in MLL2. It had been previously reported that 15% of myelomas harbor mutations in WHSC1, another histone methyltransferase, and recently a UTX point mutation had been reported, so we made a gene set of the WHSC1 family, UTX family, and the MLL family, and when you do that, you get a highly significant result, I should say, eliminating the cases we used to make the hypothesis and only looking in the other samples, and 19% of the patients have mutations here in the histone methyltransferase. Is this working through HOXA9 demethylation? Well, one way to do it would be to take multiple myeloma cell lines in which the HOXA9 is turned on and ask whether it's essential by knocking down HOXA9, and the answer is it is. That knockdown of HOXA9 in those cell lines, in fact, leads to significant decrease in viability of those cell lines. Now, these were known gene sets. What about new gene sets? Could we scan the database of MSigDB, our database of pathways, and identify new gene sets? And the answer is yes, and one comes up as highly significant. The coagulation factor pathway, the thrombin pathway. That's a little weird. Thrombin is supposed to be around to make clots and things like that. What's it doing here? And, but yet 16% of cases have mutations in the thrombin pathway here. Well, thrombin, in addition to causing clots, is mitogenic through the PAR1 receptor and possibly other receptors. And so the hypothesis here, not proven by any means, but highly significant, is that this may be acting at least locally by increasing the amount of thrombin and then having an automitogenic effect through that. Anyway, these are the kinds of things that emerge by looking at it. You get all sorts of biological insights, known mutations in genes you're not at all impressed by, KRAS, NRAS, P53, all kind of obvious, but lots of new things emerge, or deeper pictures of a lot of other things emerge. And of course, since we have 20-odd whole genomes, you could even look at non-coding regions and ask, any non-coding regions emerge? And the answer is yes. 18 regions across the genome emerge as being significant with respect to a density of non-coding mutations. 4 of these occur in genes that themselves have point mutations in their coding regions. 3 of them occur in one gene that's a GPCR, 2 of them in another gene that's a GPCR, and these 2 genes are close homologs of each other. Can't prove it's important, but it looks mighty suspicious. And there's 5 mutations that are occurring in the neighborhood of BCL7A, which is a putative tumor suppressor gene disrupted in Burkitt's lymphoma. So this may be the first evidence of non-coding mutations playing a role here in these genome-wide screens, at least. Anyway, what's to do now? Well, there's lots of hypotheses that emerge to test. By no means am I against the hypothesis business. I mean, it's a great business. But where do hypotheses come from? We're in the hypothesis-generating business, and there are an awful lot of good hypotheses. And this is a mere 40-odd samples that have been sequenced. One really wants to sequence hundreds of samples and look deeply at what's going on there. So that's one example of where genomics and cancer are talking to each other. Let me give you one more example. And this is work that will relate to something Jeannie Lee was talking about with long non-coding RNAs. And in particular, it's been collaborative work with John Rinn and Mitch Gutman and Aviv Regev, all connected with this project. And in one particular part I'll particularly note, with P53, collaboration with Tyler Jacks. So the epigenome. Jeannie was talking about the epigenome and was saying that she might be the token person talking about the epigenome. Well, I'll be another token person talking about the epigenome because I think it's an important thing. And you have all these marks on the epigenome. Sequencing has made it possible to rapidly localize those marks by using an antibody against a particular histone modification or other things, pull down all the DNA that's associated with a histone with modification, sequence it, and look for patterns of where all different kinds of marks are. Well, one of the patterns that emerges is that an actively transcribed gene has histone 3 lysine 4 trimethylation around its promoter, and histone 3 lysine 36 trimethylation along the length of the elongated transcript, the transcribed region. This is a signature of a gene. So you could start looking for genes this way. Now, why do I care? I care because there's been this controversy in the literature of genomics for quite some time about how much of the human genome is really truly transcribed. On the one hand, there are these papers that are saying every nucleotide of the human genome is transcribed. But to do that, you have to get down to very, very low levels, and it's very messy, and I don't want to think the genome works that way. On the other hand, you have beautiful work like Jeannie Lee describes with EXIST and T6, beautiful, kosher, long, non-coding RNAs that show nice properties. Some work of hot air, but there's only been a dozen to 20 really well-characterized examples of long non-coding RNAs until the past couple of years. Are there really a lot of these things? Well, chromatin signatures can tell you in an unbiased fashion. Take a tissue, make a chromatin signature, start looking at, looks like a gene, looks like a gene, looks like a gene. Is it known protein coding gene? Cross out the known protein coding genes, find new ones. When you do that across a bunch of tissues, you find 5,000 instances of long intergenic non-coding RNAs, a whole collection of these things, about a quarter as many as there are protein coding genes. They're a small portion of the genome, mind you. They occupy less than 1% of the genome in terms of the mature transcripts that are made, but they're highly conserved. Well, not as highly conserved as protein coding genes, but they're well conserved. And they're as conserved as EXISTs and hot airs and things like that. Their promoters are very well conserved, et cetera. What do? Well, take the gene expression patterns. Get the gene expression patterns of all these non-coding RNAs, in this case with about 400 of them. Compare them to the gene expression patterns of protein coding genes and start keeping score. Here's a non-coding RNA and its gene expression pattern across a bunch of tissues. Let's see how it compares to genes in the NF-kappa B pathway. Ooh, well correlated. We'll give it a good score. How about fatty acid metabolism? No correlation. Give it a mediocre score. P53, anti-correlated. We'll give it blue. And you can do that for 1,600 different pathways. And every non-coding RNA then gets some identity as to how it's being expressed. You can then cluster the link RNAs against these functional gene sets and begin to say, if I've got proteins who have known functions, I can begin to assign guilt by association functions to these non-coding RNAs, and I can find things involved in that allegedly could be involved in immune response, brain processes, muscle development, gametogenesis, or P53 related. So let's take P53 related, since this is a cancer meeting. So you can then check that these things that show across tissues correlations with the expression of P53, in fact, show appropriate induction when P53 gets induced in various different settings. And here, of course, is all collaboration with Tyler Jax's lab, where you have P53 wild type and knockout cells from appropriate mice. And you can see that they're really induced. The genes that show this induction have in their promoters, these non-coding RNAs have in their promoters P53 motifs. They look very kosher. And here's one in particular. It lives nearby the P21 gene, and we'll give it the name lincRNA P21 because it's nearby. What does it do? Well, P53 is known to activate lots of genes, and now lincP21 gets to be one of the genes that P53 activates. It also is known to repress a lot of genes. How it represses genes is poorly understood. If you knock out P53 and you knock out lincP21 separately with shRNAs, you can ask what effects. Knocking out lincP21 affects the apoptosis phenotype, but not very much the cell cycle phenotype in these cells. But when you look at gene expression, you get a very interesting clue. lincP21, when you knock out this non-coding RNA, affects a set of genes that is largely speaking a subset of the genes affected by P53, and particularly the ones that are repressed in the P53 pathway, leading to the hypothesis that lincP21 is functionally acting as a repressor in this pathway. So is it involved as a repressor, and if so, how? Well, if you take lincP21 and biotinylate it and take a control lincRNA and you incubate it with cell extract and you pull down what proteins come down with it and run them on a mass spec, you find HNRNPK. the heavy nuclear protein, or ribosomal nuclear protein K. You can show, and John Rinn in his lab particularly have shown that it binds link P21 in vivo and in vitro. It binds the most conserved regions of this non-coding RNA, and it binds directly to P53 repressed genes. You can show that the link RNA is required for the binding of the protein to these promoters, and that the binding is necessary for the P53-mediated apoptosis, at least the full measure of P53-mediated apoptosis, which presents a hypothesis that link P21 collaborates with HNRNP-K to repress genes in the P53 pathway. Role in cancer, could this be a mutation? Could this be a target of mutation in cancer? Nobody's ever looked. In our exome screens, we've never had it on there before. Now we're beginning to add these link RNAs because this could potentially play a tumor suppressor role. What about more than just this link P21 gene? Well, it turns out that about a quarter of these link RNAs bind chromatin-associated proteins. So a quarter of them bind polycomb, in fact, and about another sixth or seventh or something like that bind other chromatin-associated proteins. And so it's not unreasonable to think that this is a general theme about these link RNAs, is that a significant fraction, of course I should say, exist is a classic example, and hot air is a classic example of non-coding RNAs binding, but now this suggests that perhaps a quarter of that whole universe are involved in that way. And you can functionally test that across the board by asking, if you knock out polycomb and you knock out the link RNA, which genes are dysregulated? We did that for the P53 link P21 case. You can do it for a bunch more cases of link RNAs associated with chromatin proteins. You can look at the gene expressions. You can say, are they similar or are they what? And I won't go into the details. And the short answer is the majority of link RNAs that are associated with chromatin binding proteins, when you knock them out, have gene expression patterns that are related to, in a highly statistically significant manner, the gene expressions of knocking out those chromatin binding proteins. So the hypothesis is that there are a large number of non-coding RNAs that function as repressors, perhaps in different transcription factor pathways. They collaborate with different proteins to localize in ways still mysterious to us how this gets done, that they get to the right place, but act as repressors. And there may be a whole layer of interesting regulatory biology that sits there, and at least with the P53 example, potentially has implications for cancer. I'm going to, in the interest of time, skip over the last bit, which was going to relate to Jeannie's high 3C, but no time for that, and simply say, well, maps can teach you lots of things. There's lots of things still to learn about cancer, and I think the collaboration between cancer and genomics is about to just skyrocket because the costs of producing data are dropping so rapidly that I will predict that within a few years it will be a routine thing for anybody characterizing any cancer model in a research laboratory to at least get the whole thing sequenced across its exome and pretty soon after that across its whole genome. And I would hope that at some point within the decade to come, it'll also become a pretty routine thing in clinical laboratory settings as well. Not that it will immediately answer everything, but it will provide us with a plateful of hypotheses and a tremendous amount of data, some of which will be clinically useful. I just want to stop by acknowledging the wonderful collaborators on the broad topics of genome sequencing at the Broad and cancer genomics generally, but particularly the multiple myeloma project, on which Todd is the senior author of this paper, and Mike Chapman and Mike Lawrence, and the LinkRNA project that's been an ongoing collaboration amongst John Rinn, Mitch Gutman, Aviv Regev, and then particularly with this new paper that is in press now with Tyler Jacks, and I won't have talked about that last topic there, and I will stop.